2. Diagnosis and genetics. Diagnosis. Biedl et al. 1999 and 2001 suggests that the presence of 4 primary features, or 3 primary features plus 2 secondary features, is necessary for a clinical diagnosis of Bardet-Biedl syndrome. Primary features: rod cone dystrophy, polydactyly, obesity, learning disabilities, hypergonadism in males, and renal anomalies. Secondary features: speech delay or disorder, developmental delay, brachydactyly, polyuria or polydipsia, ataxia, poor coordination, diabetes mellitus, left ventricular hypertrophy, hepatic fibrosis, hypertonia, and hearing loss. Genetics. In most cases of BBS, both parents carry a normal gene and a faulty recessive gene. Although the parents have one copy of the faulty gene and are called carriers of the disease, they are unaffected by the presence of the faulty gene. For a recessive disease to occur, a child has to inherit 2 faulty copies of the gene, one from each parent. The child from each pregnancy has a 1 in 4 chance of being affected. If the child is not affected, then there is a 2 in 3 chance that they will be a carrier of the faulty gene for BBS. Some BBS genes are more common than others, and there is variation in the frequency with which they are being carried, known or unknown in the population, and across certain ethnic groups. For the most common BBS genes, BBS1 and BBS10, the frequency is estimated to be 1 in 250, whereas for a rarer gene such as BBS9, the frequency is closer to 1 in 820. The risk of encountering another carrier increases if couples are related. To date, 2025, mutations in over 25 BBS genes have been identified in 80% of BBS patients. It is thought that there are still more genes to be discovered, since not all patients have an identified mutation in any of these identified BBS genes. Some genes are more common than others. Around half of patients in the UK have mutations in BBS1, and a quarter have mutations in BBS10. However, patients who carry mutations in the same BBS gene can display quite different symptoms of the condition. One might have extra digits at birth, whereas another person with an identical mutation may not have extra digits at all. It is hoped that the comprehensive genetic testing currently available will improve our understanding about the progression of BBS in the future, and whether any particular aspects in some forms of BBS may need closer monitoring. Cilia. Although the mechanism that leads to BBS is still unclear, the main cause of the symptoms that feature in BBS is due to abnormally functioning of structured cilia, long, thin, hair-like projections that stick out at the surface of a cell. There are 2 types of cilium, motile and non-motile cilia, also called primary or sensory cilia. Motile cilia are present on cells where their movement helps to perform an essential function, for example in the respiratory tract or the middle ear. These cilia can be affected in some people with BBS, causing respiratory difficulties or sensory neural deafness. More information can be found in the Additional Features section of this booklet. Non-motile cilia are found on nearly all cell types. Their main function is to receive signals from outside of the cell and transmit them to the inside, allowing neighbouring cells to communicate with each other. They play an important role in terms of smell, vision, touch, and temperature sensation. They are present in the retinal photoreceptor in the eye and in kidney cells. Mutations in BBS genes cause changes in the proteins that are needed for the current functioning of cilia. And for this reason, BBS has been categorized medically as ciliopathy. The scientific community is trying to gain a better understanding of exactly what roles the BBS proteins play in cilia formation and function. Carrier testing. Where BBS is confirmed genetically, a simple carrier test is possible in at-risk adult relatives, for example siblings, to help determine their own risk of having affected children. Knowledge of the precise BBS mutations can also provide the basis for prenatal screening tests, should parents need to know in the early stages whether a pregnancy is affected. Quote, "During the early days of diagnosis, it is important to allow time to come to terms with all that has happened, and to give yourself time and space to adjust." End of quote, parent.